Hello, I am Seth Stanton, and today I'm going to teach you how to catch your evil twin.
Identical twins have essentially the same DNA sequence, making the identification of the forensic evidence they leave behind extremely difficult. Like in the murder and hijacking case in Atlanta, Georgia that led to the death of a schoolteacher, the police ran a DNA analysis off of a cigarette that had saliva on it and found one Samuel Stanton's DNA. After he was convicted, the authorities discovered he had an identical twin brother, Seth Stanton. After getting this information, they ran a fingerprint analysis of both twins for fingerprints that they had uncovered on the steering wheel of the victim's car. The results concluded that Seth Stanton was the actual criminal. The only reason they didn't convict the wrong twin was because of fingerprints. But how do you tell the difference between identical twins in case of a setting that only has DNA evidence?
Researchers at the University of Huddersfield recently developed a cost-effective and accurate method for differentiating between the genetic profiles of identical twins. This method looks at DNA methylation, a biochemical process that helps manage gene expression, which means turning genes on and off. As identical twins age, different environmental factors affect their genomes, or the ways in which the genetic material is expressed. These differences can be seen in their corresponding DNA methylation. Researchers developed a way to compare DNA methylation using a process called high-resolution milk curve analysis. What HRMA does is to subject the DNA to increasingly high temperatures until the hydrogen bonds break. This is known as the melting temperature. If one DNA sequence is more methylated than the other, then the melting temperatures of the two will differ, a difference that can be measured. This will establish the difference between the identical twins. The DNA sequence can also be analyzed for mutations and compared against each other, but this process is time-consuming and expensive. Eurofins, the European leaders in genomic services, forensic science, and paternity tests, have successfully completed a genetic and forensic search project on twins. Genome sequencing is a technique that allows researchers to read and decipher the genetic information found in the DNA. The vast amounts of data collected in the process are then analyzed by means of intelligent proprietary information technology called bioinformatics. Here's an example of how this data, or the DNA sequences, are collected by bioinformatics.
Have thousands of replicas of each of the genome pieces, all with the same sequence of A's, T's, G's, and C's. But we have to read them all somehow. To do this, we need to make a batch of special letters, each with a distinct color. A mixture of these special colored letters and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome. This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book written in a language you don't speak. You can recognize all the letters, but still have no idea what's going on. So the next step is to decipher what the sequence means, how your genome and my genome are different.
Eurofin scientists investigated the genetic material taken from the sperm samples of two twins and from the blood sample of child of one twin. Bioinformatics analysis revealed 5 mutations. These 5 mutations are also called single nucleotide polymorphisms. They were present in the twin father and the child, but not in the uncle twin. These findings prove the hypothesis that there are rare mutations that will occur early after or before the ovum has split into two, and that such mutations will be carried on into the body and sperm cells. These are the 2 techniques that help define the differences between identical twins. Since then, some criminal cases have been reevaluated so the authorities can conclude that they have the right identical twin. Thank you for your time. I hope you enjoyed my video.